Okay, can you hear me? Is the video still glitching really bad? Alright, so let's try this. Let me know if it starts glitching really bad, but y'all's comments are about 30-second lag to my video, so. Grayson, you're an odd duck. Okay, sorry guys. I'm trying to look through some of y'all's comments. Okay. Only those legit questions will I actually answer. A couple of y'all aren't logged into a name that I actually recognize, so I'm not even going to talk to y'all. Okay, so Kyle Benar, not mentioning or talking to you. That Mark guy, not talking to you. Pancake, you're Elvis, okay. Alright, Elvis, yes, biochem was pretty gnarly. Alright, questions about biochem. Guys, keep in mind, so you're going to have 50 multiple choice questions on the test. They're just straight multiple choice, no completion or anything. Well, actually, let me back up for a sec. I feel like it's just going to be multiple choice. Let me think about that. If there were any completion or fill in the blank or anything like that, it'll be relatively ones that y'all should know. But yeah, okay. Grayson. Oh, yes, turn in your dog food tomorrow. That's fine, whatever. All right. So biochem. Guys, that's a whole chunk of stuff to try and go over in this amount of time. Things that you need to remember. It's good you're taking up like a fifth of the screen. Yeah, I see that on my end. I'll see if I can fix that. Okay. The biochem stuff. Like I said, sorry, I'm trying to see if I can fix my screen. I don't know if I can. Hold on just a second. There. Did that change anything on y'all's end? I don't know if the video output low. So it might be glitching on y'all right now. So sorry. Okay. Biochem stuff. So we've got everything from the 4 organic compound groups. Organic, you can also say polymers. You can say biomolecules. They are the 4 groups that you'll have to know. You have to know their names. We've studied the 4, but in different components, and they keep coming back. You know, they keep coming back as DNA, as chromosomes, as parts of the cell membrane, stuff like that. All right? So here's the thing. Biochem questions, obviously you need to know that their polymer names are the carbohydrates, such as polysaccharides. You've got to know nucleic acids, such as DNA and RNA. You've got to know lipids, right? That can come in many forms. And then you've got to know proteins. So pretty much everything that makes up your body, proteins. So you've got to know the monomers as well that go along with those polymers. So carbohydrates, you've got to know those are made of monosaccharides. You can kind of remember that they're monosaccharides because mono for monomer, that they are the individual basic unit that makes them up. And then proteins, amino acids, right? Amino acids individually will make up the proteins. And again, we'll talk all about proteins and get into depth after we come back from Christmas break. All right. Lipids, fatty acids, and glycerols. They're not real true individual monomers since there's 2 of them making them up. But you got to know fatty acids. If you remember, lipids are examples of fat, or fats and waxes are examples of lipids, then you know fatty acids make them up. And the nucleic acids, those are the nucleotides, right? PBS, phosphate-based sugar. All right, if y'all make yourselves a little chart, I'm going to kind of back up a little bit. So you kind of put polymer there, put the monomer there, put the structure, and then put examples. If you kind of make yourself a little chart like this for the biochemistry section, that'll help. That's as far as what polymers are they, right? Carbs, proteins, nucleic acids, and lipids. If you kind of just put those down, so those would be the polymers, those are the biomolecules, those are the organic compounds. Why are they organic? Because they have carbon in them. Okay. Hey guys, on the chat, so many of y'all are saying so many things that just keeps pushing. I only get to see like 10 in my screen. So if I don't get to see a question, you might need to ask it a couple of times so that it comes back in one of the 10. Okay. Know their polymer names, then know the monomers that make them up, right? So carbs are the monosaccharide. I'm just going to abbreviate. The proteins are the amino acids, AA. Nucleic acids are nucleotides. And lipids are the fatty acids and the glycerol. Okay. Then as far as their structures, remember you can always end up noticing a carbohydrate based on its shape because it will either be a pentagon or it'll be a hexagon. Can I just point out that I did a pentagon and hexagon really well from the side? Yeah, my AP bio class would be so proud. Proteins, you're not going to notice structure, like you won't see anything structurally that will stand out, but what you will see is they have nitrogen. Remember that little R group, that side chain? It makes every amino acid different. You might write down that there's only 20 amino acids that make up the human body. There's over 500 in the world, but only 20 make you unique. Okay, nucleic acids, you do remember, or you need to remember how to draw a nucleotide. Okay, you need to remember your phosphates are always circles. Your sugars are always the pentagon. Your base is always the square. So what makes you up? PBS. You learned it on Learning Channel, PBS. You got to be able to draw a nucleotide. Lipids, they can come in 2 forms. They can either be straight chains, called saturated fats, or you might see some double bonds happening, and that would be an unsaturated. Now, let me redraw this saturated. What I mean by straight is it still looks zigzag, but it's full of bonds. It doesn't have any double bonds because an unsaturated can do this. and it bends. Unsaturated fats are the ones that come from plants. We like unsaturated fats. They're considered more healthy for us in a way. Then be able to tell me just some basic examples. Carbs, sugars, proteins, hair, nails, skin, muscles, nucleic acids, DNA or RNA. ATP is also nucleic acid. Lipids, fats, oils, waxes. The most famous one that we talk about is this guy, the phospholipid, right? Makes up your cell membrane. All right? If you make this kind of basic chart, if you definitely feel you need it on your cheat sheet, then put it on your cheat sheet. I think really you could probably just make this and study it and not have to put it on your cheat sheet. Okay? But you've got to remember polymers are made up of monomers. These are your organic compounds when we talk about them. All right? Okay. Unsaturated fats. Good, saturated, bad? Yes. In general, unsaturated fats are relatively considered the healthier because we can break them down. They're plant products. They're the ones that are liquid at room temperature, such as corn oil, coconut oil, olive oil. Animal fats are like Crisco. They're solid at room temperature. Okay, it's what comes off your bacon grease and stuff. Okay, if we exempt world geography is, oh gosh, okay. Metrics. Okay, Grady, you definitely need a little bit of the metric system review. Okay, all right, you've got to remember your little acronym. King, Henry, died, by, drinking, chocolate, milk. Okay, or kind-hearted dad's baked dark chocolate muffins. It doesn't matter to me what you use. I put it in the stair-step method, so you remember that kilo is the largest, millimeters the smallest. All right? Whatever you start at, base, remember, is the 1. So liters, 1 gram, 1 liter, 1 meter, 1 second. It doesn't have, it's not a point, it's not a decimal, and it's not a tens. Base is a 1 step. Okay, wherever you start, you don't count that step, but you have to count the step you get to. So, if I had 1.4 meters, and I wanted you to go to kilometers, okay? The fact that this meter doesn't have a prefix suffix in front of it tells you that you are on base. If your unit has any kind of prefix, okay, any kind of prefix letter to it, you know you're on one of the steps. So there isn't one, so I know I'm on base. So I'm here at 1.4. I want to get to K, which is up here. So I've got to move 1, 2, 3 steps up. So that means that decimal's got to move 3 steps over, the same direction. So I've got to move 1, 2, 3. Put the decimal after you put your third step and fill those spaces that don't have a number with zeros. So that 1.4 kilometers turns into 0.0014, or sorry, that 1.4 meters turns into 0.0014 kilometers. Okay? The one that y'all usually mess up or just flip is this D or that D. This is a larger D, a larger unit measurement. So it is dA or dK. This D is a lowercase d. It is a smaller unit of measurement. It does not deserve 2 letters. So it will simply just be D. So the difference between the two would be 1 decameter versus 1 decimeter. The lowercase d does not get 2 letters. Only the larger deca gets 2 letters to its prefix. Okay? So, give you another example, Grady. Okay, so let's say I wanted to go from 25 hectometers into millimeters. So, because this is a whole number, just like in math, you add a decimal after it. There is no decimal, so you got to place a decimal. So if I'm here, I don't count the step I'm on. I count the step I've got to get to. So I'm going to go 1, 2, 3, 4, 5 steps. No, we don't count in between. That's what some of y'all try to do. Y'all try to count in between the letters. No, you actually count the letters as steps. So I've got to go, what was it, 1, 2, 3, 4, 5. So I'm going to move that decimal 1, 2, 3, 4, 5, put the decimal, fill in those gaps with numbers. In this case, because it was whole number, it's going to be 5 zeros. Okay, so 2.500,000 million millimeters. Okay. All right, Grady, I hope that kind of answers your question for that. Deca is the first D in yes in your sentence. Deca's the first one, yeah. Okay, can you post the exam schedule for the week, please? I mean, I can post it. It's also just on the school's website. You can just find it there. All right. I know some of y'all still have a lot of questions from biochem, so I can answer them. Just, you know, get specific. I did see a question about hypertonic maybe. All right. So the word tonic, remember, is actually referring to the concentration of salt. All right. It's not the water, it's how much salt. So when you see tonic, then you got to think salt. So the word hypertonic means high salt. Hypotonic means low salt. All right. So remember, in all those questions, kind of draw yourself a little picture so that your cell is surrounded in an environment, okay? So that's what this box is. It's just the environment with a cell placed in it. So if the question says that the cell is hypotonic, if it says the cell is hypotonic, then just in reference, then that means that the solution surrounding it is hypertonic. Okay, they have to be opposite from each other always. Unless it says iso, then they both are iso because iso means equal. But if one's hypo, one's hyper, always. So in this case, if I'm saying that the environment is hypertonic, then that means water's going to leave the cell. The water that is inside the cell cytoplasm is going to leave. Why? Because water goes where hyper is. Water goes where more salt is. It wants to always even things out. Water loves the salt. So now our water is going to leave our cell. Our cell is going to now shrink in its cytoplasm, so our cell will actually shrivel. If it were a plant cell can't shrivel, but what happens is that cell membrane rips away from the cell wall, causing what we call plasmolysis. The plasma membrane breaks, and it's actually breaking away, right? Now, if we think of this in the other direction, where the solution is hypo and the cell is hyper, now water is going to rush into the cell, causing the cell to expand and potentially burst, which is lysis, okay? You've got to remember what happens if it's a plant cell or if it's an animal cell. Because animal cells don't have that cell wall as a protection, then they will shrink and they will burst. A plant cell, it can tear its plasma membrane away, but it can't shrink, really. It becomes weak, right? So we see a limp, weak little plant, but then a plant also can't burst. So it becomes very turgid. Turgid, okay? Do we need to know plasmolysis? Yes, it could be potentially a word that I ask. It could potentially just be it could be an answer choice, but yes, it is part of your notes that we talked about. Can I come in tomorrow sometime during another class? No, Leilani. Tomorrow is lab practical day, so I won't be there. Really, you can't come see me in the morning. Tomorrow's not a morning tutoring. I'll be setting up for the lab practical. No lunch tutoring tomorrow. Y'all can try and catch me tomorrow after school. But yeah, you can't come in tomorrow. Tuesday and Wednesdays, when it's review class period times, if you're in a class period that you're exempt from and your teacher will let you come in, that's fine with me. But that's only on Tuesday. Actually, is it just Tuesday or is it Tuesday, Wednesday? But yeah, you can't come in tomorrow. Okay, is the term lysis only in animal cells? Yes. Yeah. Only an animal cell can burst. So plant cells cannot. or bacteria? Because they have a cell wall too. Okay, how much do we need to know on writing the chemical equations? I think you mean the photosynthesis and cellular respiration equations. You need to remember what they are, and you need to know that they're balanced, right? If you just remember one, then you know the other one. Just reverse it, okay? Think about, what does a plant need to survive? A plant needs sunlight. A plant needs water. A plant needs CO2. What does a plant make for us? They make oxygen, and they make sugar for themselves. Where do you add in the balance? Everything except for sugar, because sugar already has 6 carbons. But yes, you need to know them. There's prefix suffix, right? Yes, there will be some prefix suffix on there. It's only a couple of them though. It's only like maybe 4 or 5. I don't make much of the test prefix suffix. Okay, in cellular respiration, do the 6 oxygen in the beginning leave the carbon and go straight to the ETC, waiting for electrons to make water? I'm trying to understand where you're coming from. You're talking about, like, just at the beginning, like, here we are, you're talking about the beginning of the equation. So you're breathing, guys. We constantly have oxygen molecules floating around in our cells, constantly floating around. So yes, where is one of the primary places they'll go to? They go into the mitochondria. So they're filtered into the mitochondria. The mitochondria then help convert them into water. Yes. Can you explain passive and active transport? Okay. Savannah, remind me and I'll answer your question again about the lab practical. Guys, remember, if I don't see your question, it's because, seriously, there's so many of y'all. Y'all keep, like, pushing up, and I can't see all of them. Okay? All right, passive and active transport. Difference, energy. What kind of energy? The only kind of energy that makes sense in your cell. ATP. ATP is your cell's form of energy, right? So, passive and active, if it's a passive transport, it doesn't require the energy. If it's an active transport, it does, right? You're active. When people say you're really active, you have a lot of energy. Right? Okay, so passive and active transport. In a passive transport, you have simple diffusion. Okay? All diffusion means is that it's able to pass through the cell membrane. It doesn't need any kind of help through a protein. Okay? Simple diffusion, that'd be gas. We're talking O2 and CO2. Regular diffusion. Okay? Diffusion, simple diffusion versus diffusion itself. Simple diffusion literally means it just passes. Just gases are able to pass through that phospholipid bilayer. Now, when we talk about diffusion being part of the passive transport, we can break it up into 2. Diffusion can either be osmosis, the movement of water only. Now, it doesn't require energy, but it does require protein to help. Why? Because water cannot pass through those phospholipid tails. Those tails are hydrophobic, so water has to protect itself. So it has to pass through a protein. Then the other kind of diffusion is facilitated diffusion. Facilitated diffusion, again, it just means it needs a protein to help protect it to get through. But here's the thing. It's not osmosis, because osmosis is the only movement for water. Facilitate diffusion. This would be for proteins. Yes, proteins get help through proteins. I know that sounds weird. Sugars. Okay? Any kind of large molecule that needs to get into the cell needs help through facilitate diffusion. But again, yes, is it helping? It sure is. But it's helping without the use of energy. Okay? Active transport. Your active transport requires the energy. So, ATP. It's got to use that for energy. What are your forms of active transport? One of the examples we talk about is your NAK pump, your sodium potassium pump. This is actually for your nervous system. Okay? Other ones we talked about, endo and exocytosis. Endo and exo. We talk about a cell needing to take in a bulk amount of something in, right? So it's got to take it into the cell, or it's got to push it out of the cell. That was that little acting we did where we kind of grouped around the bacteria, we took it in, we chopped it up, we pushed it back out, okay? So endo means inside, so this means you're going to form a vesicle. The cell will actually form around something. Okay, it'll kind of get close to it, and then it will form a vesicle around it, bringing it into the cell. Okay, so it can break it down. Exocytosis. Exo, it's going to take that vesicle. It's going to fuse it with the cell membrane. I'm trying to draw this well enough. Okay, and push it out into the environment because it already broke it down. Okay, there were two kinds of endos. Endo could be phago, which means to eat. So that's taking in an actual solid substance, like a bacteria. And then there's pino, to drink. That's taking in the liquid environment because there's dissolved solutes in it. Like there's sugar compounds, there's protein, amino acids. There's something dissolved in the solution surrounding a cell, and it'll actually take it in. So this could be antibodies for a white blood cell. This could be, I don't know, clotting factors for a cell, something like that. Okay, rough ER makes protein, smooth ER makes lipids. Yes, that would be correct, Elvis. Can you explain receptors? Guys, all a receptor is a protein that hangs off the outside of the cell's membrane. And what can a receptor do? Many, many, many things. But all receptors are. They're just various forms of proteins. They could be lipids. They could be various things. So when you see receptor mediated, when we talk about an active transport, again, all that is, that's an endocytosis. It's just so specific that the only way that cell knows to take in that compound is because it actually told the receptor, hey, I'm here, I need you to take me in. Something like that could be seen in a cell taking in cholesterol out of your blood so that you don't get blood clots and stuff and clogged arteries because there's too much cholesterol. Hopefully your receptors are working correctly. Okay, covalent and ionic bonds. Okay, covalent bonds, those are simply just sharing. Why? Think of covalent as in like you're cooperating with each other. So a covalent bond would be any time two atoms are sharing the outermost electrons, their valence electrons. We see this between hydrogen and oxygen atoms making up a water molecule. So within the water molecule are covalent bonds, okay? Ionic bonds, that's when two atoms are either lacking one or two electrons on their outer shell, or they need one or two electrons on their outer shell. So instead of them sharing, they actually will give up electrons or they will accept their electrons. That's where we came up with the names, okay? Cation and anion. When an element gives up an electron, so he gives up something negative, then that makes him technically more positive, so he becomes a cation. That would be sodium, for example. Okay, sodium loves to give up his electron. Okay, an anion, you don't want to be a negative ani, right? An anion is the atom that now gained that electron. Chlorine is one of those. So for your sodium chloride salts, right, on your table right now, sodium became the cation because he was willing to give up a negative electron. And if you give up something negative, that makes the atom more positive because now it technically has one more proton than it does electrons. And so protons are charged positively, it becomes more positive. That chlorine now took on an extra electron, making its whole atom a little bit more negative. So it became the anion. Okay? Every time you have one cation, you have to have an anion. What do you recommend we study the most on? Is it all multiple choice? Are there units even for all the questions? I would not say the units are all even by any means. Oh, well, yeah, I mean, the units are relatively even. I'm not going to say it's exact by any means. I'm not overly focusing on one unit. Like, you're not going to see a ton of biochemistry or anything like that. So, yeah, they're relatively even. It's majority multiple choice. I'm still, I said it earlier in the video, I'm still playing around if I want to add a few what I believe should be very well-known terms and definitions as completions. I will let y'all know that for sure tomorrow. Okay. Oh, heavens to Betsy Davis. Dear Lord. Okay. Is y'all's video not loading? We can see it. Chelsea, let her know if she can't see it. It's probably because she's trying to get it on her Chromebook. And apparently trying to watch a live stream video on your Chromebooks is not allowed, so it restricts it. Are we going to know the shape of carbohydrates and stuff? Well, yeah. I went over that earlier, Elvis. You've got to know carbs are either pentagons or hexagons. Nucleotides, you've got to be able to draw that nucleotide, that PBS. That P for the circle, that sugar, pentagon, and that rectangular base. Do you have any kind of acronym for the characteristics of life? I do. I need to remember it for just a second. I haven't said it in a long time. Okay. Lab practical, Savannah. So the lab practical, every table will be set up the same way. So you will go to a table by yourself and you will do the things on the paper I give you. So whatever's on that table will be the same at everybody's. Okay. It's just one person will be at a table at a time. If y'all end up needing to think, I'm going to have y'all sit down, think at your table so that I can get that table open again. Okay, so the lab practical can be anything in your lab notebook. So from the very beginning, we started with lab equipment. We did sponge scientific method lab, quantitative, qualitative data, lab equipment name and functions, triple beam balance, microscopes, and it can even be, well, not even, it will be some of the labs we've done and their results. So ones that y'all have written in your lab notebook. If you didn't write in your lab notebook, then that's going to hurt you. Hopefully you still understood it well enough. The lab notebook just kind of serves as a cheat sheet for y'all during the lab practical. Will the test be Scantron? No, it will not. I'll count that as one by default. Don't know what that is. She'll tell you tomorrow, Savannah. So the entirety of photosynthesis model paper and cellular respiration model paper, do you recommend we study that? Oh, the entire drawing that we did? Not the entire thing by any means. No, you're not really going to have a diagram of that on there, but you still have to understand the steps. You still got to know in cellular respiration that you go from glycolysis, creating pyruvate from the breakdown of glucose, into the mitochondria for the Krebs cycle to produce CO2 and energy, and that energy to then be used in the ETC cristae to help make ATP and water, right? That's kind of the very, very general. If you can kind of tell that general, you're pretty good. If there is an oxygen, then the pyruvate stays in the cytoplasm and turns into alcohol or lactic acid, depending on the kind of cell you are. For photosynthesis, again, be generic. Light rays of photons come into the thylakoid, get the photosystems, right, excited because they have chlorophyll. Electrons then travel through the ETC of the thylakoid membrane, help create energy in the form of NADPH. Hydrogens get pushed into the thylakoid. They get too crowded, so they fall through the ATP synthase, creating ATP. That energy of ATP and NADPH then travel into the stroma. Well, they're already in the stroma. That's where they got created. So inside the stroma, CO2 is then converted into glucose because it uses the NADPH and ATP that were created. All right, if you can kind of cover back and forth, like just make a small little chart of photosynthesis and respiration, that would be good for you, right? We did it in class too. So photosynthesis and cellular respiration, you kind of make like a little chart about the where they take place, the what they use, and what they make. That'll help. Okay, so talk about the where, so thylakoid membrane, light reaction, stroma, dark or light independent reaction. Actually, I'm going to say Calvin cycle. Okay, and then break down the light and the Calvin of what they use and what they make. Do the same thing for cellular respiration. I'm not going to make it all right now because we did it in class. It is out there somewhere in some of your notes or reviews. That way I can get to some more questions because I didn't even get to eat dinner. I'm sitting here still waiting to eat my dinner and my stomach's starting to growl. So let me know if you can hear that. Fritz, I don't know what 11 and 12 is. I don't have my test review pulled up. You need to kind of type it in and tell me what that is. Trying to see if there's any questions. Do we need to know much about the difference between... I'm trying to see what the rest of that. Different fibers, like microtubules. No, you don't have to know all the different kinds, like microtubules, microfilaments, all that kind of stuff. No. Do you need to know that spindle fibers are microtubules? That would be good to know, right? The things that attach to the centromeres, that attach to the kinetochore proteins, those are microtubules. That'd be good. It is tomato soup and sandwich because I did not feel very well this weekend. Wah, 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 wah. Can you explain the enzyme and its parts? Yes, Elvis, I will. All right. Enzymes, guys. You gotta love enzymes. They are going to haunt you, so you need to know them, right? You need to remember an enzyme is a protein, okay? Not all proteins are enzymes, but enzymes are proteins, okay? You gotta remember the job of an enzyme, right? If we kind of look at your graph, okay? Remember these graphs? Yeah, you gotta know these graphs. I thought tomato soup was called spaghetti sauce. Heavens to Betsy Tucker. If animals didn't want to be eaten, then why are they made of food? Very, very good. What do we need to know how to draw? You don't need to know how to draw anything necessarily, Davis. The one thing I would say y'all definitely need to be able to do is identify the cell and plant. The plant cell and animal cell. Heavens, Betsy, it's so late. Okay, remember these graphs. Y'all got to remember which one's which. Yes, I can tell you that. If you don't remember, you have a hard time with this, maybe this is a very simple thing you could put on your cheat sheet and won't take up much space. Okay, these graphs are actually telling you your reactants and your products. Your reactants and your products. And it's telling you their energy. That's what the Y is. It's your energy, okay? This is just the progression of the reaction. The hill. Anyone remember what the hill is? The hill, the hill. The hill is the activation energy, okay? Enzymes will change that activation energy and make that hill smaller. Why? It lowers the energy needed to get a reaction going. Notice the enzyme didn't change where you started and where you ended. It doesn't affect the reactants, and products. It simply just makes the hill lower. If you were to draw an enzyme, it does not matter what you draw for your enzyme, okay? An enzyme is simply just a large protein, usually multiple proteins put together, substance. An enzyme will have a location on it called an active site. That active site is for a substrate to bond. When that substrate bonds to the active site, okay, it will now be converted into whatever it's supposed to be. That substrate could actually be two things, and they're bonding, the enzyme's bonding them together. Either way, eventually it's going to put out some kind of product. A substrate will turn into a product. So in an enzyme, the reactant is the substrate, and it turns it into a product, okay? Not every reaction has an enzyme, but when a reaction uses an enzyme, it can happen faster, because that activation energy was lower. The speed of the reaction is sped up because of an enzyme. The activation energy is lowered because of an enzyme. Okay? All right. Do we need to know the different scientists that studied the cells? Yeah, in general. Okay, Eli, you're not getting 100s, dude. I mean, yay, I'm glad you're watching, but you're not all getting 100s. Okay, do we need to know the types of scientists? I'm trying to read. The scientists who study the cells. Yes, it would be good. But quick reference, small, like nothing big, right? Leeuwenhoek, animalcules. Okay, he's the one who saw protists but didn't know what they were. Hook was the first one to identify cells, so Hook, cells. Leeuwenhoek, animalcules. Schleiden, plants. Schwann, animals, kind of like, sounds like swan. Virchow, cell theory. Yes, you do need to know the 3 parts of the cell theory. If you can't remember them, you might need to write them down. Hopefully you can remember them, because I feel like that's kind of a waste of space on a cheat card. Okay, the 3 cell theory parts. Cells, basic unit of life. All things are made of cells. Cells come from existing cells. Ding, ding, ding. Okay. Can you explain cyclins? Guys, all a cyclin is a type of protein that is continuously inside your cell and goes through moments of high concentration and low concentration in a cell's life. When the cyclins are up, that means the cell is ready to go through mitosis. If cyclin levels are down, the cell's not ready to go through mitosis. Okay, what are the effects on a cell if the enzymes die again? Okay, so remember, enzymes don't ever die, right? Enzymes become denatured. They become unfolded. So it's like, take a piece of paper, fold it up, then unfold it. It's still a piece of paper. The enzyme is still there. It's just not the shape it is, or it used to be. And now a substrate can't bond to it. But when the temperature is right, when the pH is right, it'll fold back up, take its original shape, and now the substrate can bind again. Examples of when your enzymes denature, it wreaks havoc on your body. Like, that's why you can't have a high temperature for too long, or a high fever, because literally the enzymes in your body are going to stop working. I'm not reading that aloud, Davis, dear Lord. Okay, if all cells come from existing cells, how did the first cell get created? Ooh, that's a big, long question and discussion. Not for today, Tucker. Shut off for the question, but I'm back now, so no worries. Or, okay, do we need to know a lot about tonicity? You've got to know hypertonic and isotonic. I'm going to give y'all examples. Like, they'll be worded out. I'll explain that if a freshwater cell, such as a paramecium, were placed in a saltwater environment, what would happen to the cell? Like, that one's a very simple one. So I just said that the cell was freshwater. Okay, guys, on those tonic questions, you've got to draw it. I'm telling you, drawing it helps. If you don't draw it and you miss it, I have no sympathy for you. So draw the beaker, draw the cell. I said that the cell was freshwater. Freshwater, in almost all cases, would be considered hypotonic. And saltwater would be considered hypertonic. So once you have that drawn out, water will always go where there's more salt. So more salt is outside of the cell, so water from inside that paramecium is going to leave. So that paramecium is going to shrink. It's going to die. Okay? That's why mean people put salt on snails. You're actually killing the snail. All the water is leaching out of that snail, and he is drying out to death. Do we need to know about the acidity and basicity? Okay, yes, acids and bases. You've got to know your pH scale, 0 to 14. A always comes before B in your alphabet. Same thing goes for your pH scale. 0 to 14. 7 in the middle. Acids come before bases. You got to remember that. Acids have a high amount of hydrogen ions. Boo. Bases have a high amount of hydroxide ions. Okay. Do we need to know all the different forms of ATP, like NADH and all the other ones? So ATP and all of his cousins. I would say you need to recognize them as components in questions and maybe the answer choices, but more like, you know, electron carriers in the Krebs cycle would include NADH, FADH2, and ATP. So yes, you need to be able to recognize them, but I wouldn't go too heavy on them. Like, don't lose sleep over them. Can you go over the Calvin cycle if you haven't already? What are y'all doing? Playing a game at the same time as this review? Am I not entertaining enough for y'all? Sheesh. Okay. when do you want me to take my test that I missed on Thursday? Oh, yeah. Oh, Miss Chelsea. It's you and just like a couple others. Find it kind of funny that you and Davis both missed your test on Thursday. Interesting. Yeah. You will be taking it after your lab practical. Ooh, yeah. Like, there's really no other time for you to do it. I'm pretty sure 7 is just neutral. Yes, it is. 7 is neutral. I'm actually playing... Oh, okay, Overwatch. Is Overwatch that video game where they're kind of like cartoonish, kind of like Call of Duty, kind of not? I think that's the one my son has. What were they doing? I know, exactly, Tucker. Exactly. Okay. What was the question? Calvin cycle, Elvis. Kelvin. Oh, Calvin, not Kelvin. All right, Calvin cycle. If I had a plant, I'd name him Calvin because that's what happens in photosynthesis. All right, so remember, woo! Chloroplasts. Chloroplasts have stacks of what? Thylakoids. Surrounding the thylakoids is the stroma. Outside of your thylakoid, right, here are your proteins, here are your photosystems, here's your ATP synthase. ATP and NADPH get created in the stroma. They will simply just travel to a spot in the stroma. So they were just created here. So they're going to travel over here, and they're going to get thrown into this whirlwind called the Calvin cycle. What's the Calvin cycle doing? Taking CO2 that the plant took in through its stomata, and it's going to simply convert that CO2 into C6H12O6. How does it do that? Because it uses ATP, breaks it down into ADP, and it uses that NADPH and breaks it back down into NADP. Okay? That's really all you need to know for the Calvin cycle. How long are you doing this for, Mrs. Good? And I'd like to say roughly an hour. I really would like to eat. Drop bombs like they're coming from a cherry tree. I don't even know what that means. Okay, why X-Files theme here? Never got my acronym. You did not. Trying to remember it. I haven't used it in so long. What be pyruvates? Sorry. I'm getting crap on my phone. Okay. Nate, acronym. Let me think of it. I promise I won't forget. Let me just think of it for a second. All right. Pyruvate. That's the 3-carbon molecule that glucose got broken down into during glycolysis. All right. Hold on. Sorry, my phone's blowing up. Okay. So the pyruvates, yes, you just need to know. I find it funny that Davis and Chelsea both left when Miss Good exposed them. Ah, funny. Okay. Davis is still there. Do we need to know binary fission, budding in detail, or just simplified version? Simplified version. That binary fission and budding, those are examples of asexual reproduction. They can't be called mitosis, right? Mitosis is asexual. That's how our cells do that. But they can't be called mitosis because that is the actual division of a nucleus. So binary fission, bacteria. Budding, sponges, hydra. Okay. Still here. Yay! Y'all are both still there. Okay. All right. Trying to remember. Okay. Chelsea, I love you. Who's Marokik? Who? On the review said that DNA is not replicated during SF interphase. Was that a mistake? Heavens to Betsy, if it says that, yeah, fix that. Are you sure you're not looking at meiosis 2, like it was a question about meiosis 2? That there is no replication? Oh, that's Maddie. Okay. I have more Post-it notes than Bruce Almighty right now. Good. Okay, Nate. No, it was on the review as false. Interesting. Trinity, I will definitely look at that again. If just remind me in class tomorrow to say something. Guys, we know DNA is replicated during the S phase. I will definitely double check that review. That sounds weird. Okay, Nate, your acronym, characteristics of life, something that can help Mrs. Grin sometimes is used. I want to say we can sometimes add Mrs. C. Grin there, like Mrs. C. Grin. But I don't use it all the time because honestly, the reason I don't like to use it is because this acronym uses slightly different words than I would. So it's just a little odd to me sometimes, right? And I don't like what they use for the M. This is like seriously like one a middle school 7th grade teacher would use, and I don't like it. So they will put M here for movement. You don't have to be able to move to be considered alive. Hello, coral is alive and it doesn't move. So I don't like that one. And trees don't get up and walk. So don't really like this, but whatever. Davis has left the chat. Why did Davis? What could be 30-year-old from the dark web? Well, heavens, Betsy. Okay. The chat. Okay. So instead of movement, I put metabolism. Okay, because you have to have some kind of metabolism. All right. Reproduction. Okay, obviously. Stimulus. You have to be able to respond to your environment. Okay. Growth. You have to grow. You have to blah blah blah. C, cells. You have to be made up of cells. Okay. Let's see. Evolution down here. Trying to remember. I don't see this one. Sometimes will be saved as the respiration. See, this is why I don't like this one, because it uses respiration when I would say homeostasis. Like, you have to maintain homeostasis, so I don't really like that one. So maybe I can switch these letters around. I haven't used an acronym for characteristics of life in a long time. And then the N down here, this is where they normally would fit in the nutrition one, which is weird. So actually down here, put nucleic acid. Because you have to be made up of, you have to have some kind of nucleic acid component. So that's 1, 2, 3, 4, 5, 6, 7, 8. There you go. Mrs. Seagrin. Oh, it says false because it says prophase. Oh, thanks, Trinity, for clearing that up. All right, so Nate, there's your acronym. Mrs. Seagrin. Metabolism, reproduction, stimulus, cells, growth, respiration, replace that with homeostasis, evolution, nucleic acids. So maybe we can say Mrs. C. Hinge. I don't know. Put something else in that. Okay. All right, so that was characteristics of life, a little acronym. Davis, answer your text. That's just funny. Can you explain oncogenes, tumor suppressor genes, and when they get mutated? What's a fact? What's a theory? Okay, so I'll answer those two. Oncogenes. The word onca actually means cancer, so it's a gene that will cause cancer. We all have these genes in us, unfortunately. We have these. It's just, did you inherit the mutated one? So an oncogene, we want always turned off. We want that gene not to be expressed so that you don't get a cancerous gene, okay? Tumor suppressor gene, it does that job. Its job is to suppress the tumor, stop it from actually going anywhere, stop that cell from going through mitosis. So a tumor suppressor gene, you actually want always turned on. You don't want it to get mutated. So if a mutation happens, what that means is an oncogene gets mutated and turned on, so that way it causes cancer. Tumor suppressor gene gets mutated, gets turned off, so it can't actually do its job and suppress. What was that last one that I said I'd answer? I just never... Oh, fact and theory. Facts have substantial evidence and are always the same, and they are not being altered. So gravity is a fact. It is the same no matter where you go. Gravity exists at a 9.8 meters per second squared on Earth. Okay. Theory of evolution, right? Even though it's highly supported, because we don't have substantial evidence for all components of it, we can't call it a fact. Okay, and a theory is always changing. Okay, Mrs. Good, scroll up. Why? Malignant spreads, benign stays in place. Yes. Fermentation, please. Okay. Sorry. I'm going through. She asked me who I fought. Who you fought? Did you get in a fight, Davis? Is that why you were not at school Friday? Shut your mouth. Are you talking to me, Davis? Heavens. I know what facts and theories are. I was kidding. Oh, okay, good. Barnett, leave. Okay, stop, leave. What's the thing that happens in diabetes? Cells not letting insulin in. Exile anyone who argue about the state of water. Okay, cannot see, drive. Okay. All right, so fermentation is the first question, and then is what was that last question you said? The diabetes, what's the thing that happens when with diabetes and the cells not letting insulin in? Guys, so all diabetes is, it depends on what kind of type you are. Type 1, your pancreas doesn't even make insulin anymore, so you've actually got to take insulin shots. Your cells still accept it, but your pancreas doesn't make it. Type 2 is the reverse. Your pancreas does just fine, but your cells have been so overloaded with it because of your diet that they say, stop giving me insulin. So it's two different kinds. Okay, fermentation, real quick. So fermentation, we only talk about two types. There's tons, but we only talked about two. Alcoholic, does not happen in your body, can't, happens in bacteria, yeast, that kind of thing. So alcoholic makes alcohol and CO2. Some of y'all forgot that. Okay, and CO2. That's what y'all were collecting in y'all's fermentation lab. All right, I know it didn't do as fast as we needed, but that's okay. And then fermentation lactic acid is what happens in your body. You will produce lactic acid. Yes, prefix, suffix is on the exam, Chelsea. Not very many, only about like 4 or 5. Okay. So here's the story. I threw a ball at a guy because he was trash talking. Then I ran at his friend and shoved them. I got ejected from the 7-on-7 game when he said water was wet. Then I said shut your mouth. Okay. Whoo. All right. Just making sure, Davis. Okay. Let's not get thrown out. Can you talk about stem cells, and do we need to know the different types and description? Write a fan fiction about cells. That would be funny. Okay. If you think that's funny, Jefferson, just wait until we get to protein synthesis second semester. Yeah, that one's a fun one. Stem cells. As far as detail, not really too much detail by any means, Joe. So I wouldn't be too stressed about that. Stem cells, they are what we would essentially call like a baby cell. They haven't been told what they're going to be, one way or the other, right? So there are 3 kinds of levels of stem cells. I guess you can kind of think of it like a football team. You know, you have different strings. One's more determined than the other, like as far as what their job is. So stem cells, you have your totipotent. That's the actual placenta and the entire embryo. We don't create nor make nor use or anything totipotent stem cells. We just can't. Pluripotent, That's the one we want to use. That will create every component of the embryo from scratch. That's the one we want to use. That's the one that we currently are making, the stem cell spray gun for burn victims with. Multipotent is what you have right now inside your body, okay? But they are specialized at this point. They are stem cells that will only make the different blood cells, red and white. They are muscle stem cells because your muscle cells don't divide, so you keep making more muscle cells from the stem cells, okay? Stem cells, please. Again, that's all you really need to know, okay? I'm just going through the comments real quick. You didn't even throw a punch week. No, I would have gone through an out, blah, blah, blah. It sounds like second semester is more packed than the first. In a way, kind of. But I feel like there's easier components in it. But yes, especially right there at the end, we pack a lot of learning in. In all reality, people have asked, do we cover the entire book? It's like 50-something chapters, and we're only on chapter 6. And yes, actually, we really do, in a way, cover all the chapters. Isn't that nice? But the last, 20 chapters or so are all body systems and all the different kingdoms. And we don't go into depth in all of those, but we do kind of squish them in. Okay. And not prepared for that. If you ever get, okay, who cares? I don't care about all the violent stuff. Okay. What be a parent square Punnett square, sorry. What be a Punnett square? Is there a 64 by 64 Punnett square? We want to never create one, but could you mathematically make one? Yes, you can. And I think that'd be fun to do in AP Bio. Can you go into depth on enzymes? I'll do another quick one on enzymes. I already did enzymes though, Fritz. So really just this video you can actually watch again later. Punnett squares I'll look at again. Summarize scientific theory. Already kind of did that. Okay. Punnett squares, come on. Pancake, Elvis. You know what a Punnett square is. I shouldn't have to cover a Punnett square. But I will tell you, on the test, some of y'all still kind of messed up on the terms monohybrid and dihybrid. So make sure you'll understand that correctly, right? Monohybrid, that's just the 4 Punnett square. You're looking at one set of letters. You're looking at A and A. You're looking at B and B. Okay, that's monohybrid. You're looking at one trait. The dihybrid, that's when we do the big box, right? We don't have to really do the big box because we know how to multiply fractions, right? Dihybrid is when you're looking at 2 separate traits, okay? 2 separate traits, that the A's, the B's. The R's, the Y's, whatever. Okay, good. I'm glad you were kidding. Is the review due tomorrow? I don't take up reviews, people. Remember that. Can we go over diploid haploids again? Okay. Your meiosis, guys. If y'all got confused with meiosis, you might want to draw this on your cheat sheet. Okay. So the first cell in meiosis is really a somatic cell. Okay. So that would be a diploid. That would be 2N. From that, we'll make 2 cells. This would be meiosis 1. That will end with haploid cells. Then both of those will then branch off into their own haploid cells. Okay? This is meiosis 2. We call these daughter cells. Heavens to Betsy. Okay? Those daughter cells will become your gametes at that point. Okay? So you went from a homologous set of chromosomes to splitting those homologous chromosomes. So that's why they got halved already. And then these chromosomes that are simply just replicated chromatids, their chromatids get split. And that's why it stays as a half. Because you counted these as 1, count the chromatids each as 1. Okay? Scientific theory. What do you want for scientific theory? Any scientific theory simply is a scientist has a question, they have substantial evidence to support their theory or their claim, and they have proposed that scientific theory of evolution. I don't know. lot that have turned into facts or scientific laws at this point. Okay, any kind of scientific theory can be explored through an experiment and can be explained and supported through scientific evidence due to that experiment. Okay, when you're done, can you put it up so I can screenshot? I don't know what you mean. Law of segregation, independent assortment. Okay, guys, law of segregation, independent assortment. Those are two things that Mendel's experiments helped to explain, and that later on we were able to put into place in meiosis as well. So, both of those are explaining how your gametes, your eggs and your sperm, they're all haploid, right? Every single sperm, every single egg will end up with one copy of every chromosome, which means one copy of every gene for you that makes up your body, okay? But then the law of segregation is how that happened. The law of independent assortment is how that happened. So both of those laws take place in meiosis. Segregation is the very end. Segregation is what's going to split chromatids so that alleles for genes separate. So if you're heterozygous, right, those will split, right? So one copy went over here, one copy went over there, chromatid in this homologous, chromatid in that homologous, okay? Law of segregation is when one trait's alleles separate. They're in meiosis 2. From the homologous chromosomes, they're separate, and then those alleles separate. Law of independent assortment is earlier in meiosis 1. It's when homologous chromosomes, right, they carry their own genes and traits. And so what it means is more than one trait. Segregation is just one trait. Independent assortment is when more than one trait will assort independently from each other, meaning the traits and genes on chromosome 1 from mom do not influence the genes and traits on chromosome 13 from mom. They are separate and will go into gametes separately from each other. Law of segregation is easily explained by a basic Punnett square. How the mom's alleles and the dad's alleles create an offspring. That's law of segregation. Law of independent assortment is what's explained by the big dihybrid. The fact that round seeds and yellow colored seeds did not have to go together. That they do not have to be in the same offspring every time. Okay. Okay, Mendel's laws, right? Yes, law of segregation, law of independent assortment, and law of dominance. Those are all Mendel's laws. If you need okay. All right. I don't know, waiting for some other general questions from y'all. So, yes, study some prefix suffix. I wouldn't be too highly concerned on prefix suffix because you've seen them a few times and now you're starting to recognize prefix suffix in some of your vocab and stuff, so I wouldn't be too concerned with that. Things that I personally would put on my Punnett squares. Somebody asked earlier about drawings and stuff like that. I wouldn't waste time on drawing out the big photosynthesis, respiration stuff. Don't draw that stuff. Would I draw a plant and animal cell? Yeah, maybe. Especially if you have a hard time recognizing their pictures, I would draw that. I would draw some of the charts, you know, like the organic compound charts, the photosynthesis respiration chart, basic meiosis or mitosis. If you need something like that, like acronyms, IP on Mat C, you know, kind-hearted dads bake dark chocolate muffins for the metric system, Mrs. Seagrin for the characteristics of life. If you need acronyms, that would be a good spot because those won't take up much space at all. Okay. ETC and movement of electrons. Okay, I'll describe that. Do we need to know, how much do we need to know about ATP each molecule has? Do we need to know how much ATP each molecule? Probably not. I probably won't ask the question right? Sugar gets you approximately 34, 38 ATP. It can range. That's why some people have a higher metabolism than others. It's your lipids. Those get you 146. Wowzers. That's why your body is going to start converting lipids into energy and breaking those down before they'll ever go to muscle, because you'll get more energy from your lipid breakdown. Okay. Okay, ETC, movement of electrons. I would maybe draw this very generically on your cheat sheet if that was a hard concept for y'all, the ETC. That might be a good thing to have on there. Okay. So very generic drawing, right? You have your phospholipid bilayer. Now in reality, we know that this actually looks like this, right? Phosphate heads, the tails, right? Okay. We know that your phosphate heads are hydro, right? Meaning water. Philic. They love it. Okay. Why do they love it? Because they're polar. Why? Because polar bears swim in the water. That's why water's polar. The tails are hydrophobic. They fear it. Why? Because they're fat. Oil doesn't mix with water. Kind of, that just generically makes sense. All right. So your ETC, draw your membrane very, very simply. You have your proteins. And then here on the end, you have your ATP synthase. So electrons will travel. They'll always come out at the end. Hydrogens will build up. And they will always travel through the ATP synthase, helping to convert ADP into ATP. This is generic. Now, is this cell respiration or photosynthesis? That's where the differences are going to come in. If it's photosynthesis, this is photon energy, comes into your photosystems, breaks water. Oxygen leaves. These electrons create NADPH. Right? If it was cell respiration, okay, here it's actually going a little bit backwards. Okay? Here NADH gets broken and FADH2 gets broken. Electrons will travel, but here water will be created. Okay? Water gets created. Why? This is all in the matrix. So very, very generic ETC. Okay, y'all better be liking and subscribing, also turning on that notification button. Mrs. Good, give a shout out of my channel name and tell them to sub. I don't even know what sub means. Oh, subscribe. They can see it for themselves. Okay? There you go, guys. Look at Davis. Look at Davis on the chat. Look at Davis. He wants you to subscribe. Subscribe to Davis. All right. So things on the test. It is the entire semester, right? So that's scientific method, right? Independent, dependent variables, control groups, constants, data if it's qualitative or quantitative, right? You've got to remember those. Then we went into biochem, right? Water's properties, cohesion, adhesion. Cohesion, cooperating, that's water to water. Adhesion, when it connects to something else like the side of your glass, right? Both of those, that's pretty convincing. I better do it. Oh, good. Yes. Subscribe, Elvis. Do it. Okay. From biochem, then you went into enzymes, right? Remember the enzymes? Oh, we didn't even say that on those graphs. Let me go back through those graphs real quick. Okay, I drew these, right? Right? Your basic graphs when it comes to enzymes. Okay? All right, you've got reactants, you got products, you got reactants, you got products. You got to remember which one's endo, which one's exo. This would probably be good to go on your cheat sheet. If the energy level of your products is lower, this is exo. If your energy level of your products is higher, that's endo. This question came up earlier in the chat. Anabolic and catabolic. Catabolic, think of catapult. The job of a catapult is to break things. That's what happens in a catabolic. It's breaking down. Okay? So exo would be catabolic. because your reactants literally broke down into products. Endothermic or endergonic, you're putting energy into it. That's because it's anabolic. Anabolic steroids help build up muscle. So here you got an anabolic reaction where reactants are building up into products, right? All right. From biochem, then we went into cells, so cell structure. Again, if you don't feel comfortable with cell organelles, what they look like and where they go, you might kind of draw a generic picture and what their jobs are. Okay, sorry, my phone's blowing up again. And then from cells, we went into cell cycle, so IP on Mat C. Remember your cheer. I, synthesis. P, M, A, T, cytokinesis, right? Remember that. Activation energy is the hill, right? And that's what enzymes do. They lower that activation energy so the hill is smaller so they can get through it faster. My son plays Xbox. All right? And then from cell cycle, right, things you got to remember. I would probably, if you need it, if you don't know it, but if you need it, draw your chromosome, right? Label your chromosome. That's the centromere, your sister chromatids. 2 of them would be homologous chromosome. Kinetochore proteins build on the outside of them. They can actually come together and touch, called synapses. And here they might potentially cross over. When would crossing over happen? Prophase 1. Right? You might draw your chromosomes if y'all don't really remember it too well. All right. Your angry. Okay. Then from mitosis, we went into meiosis, right? Production of sex cells, also known as gametes, eggs, sperm. Diploid go into haploid. Homologous chromosomes separate, and then sister chromatids separate. So in a human, we go from 46 to 23 to 23 again. And then for meiosis, we went into Mendel, right? You just took your Mendel test. Yes, Mendel questions will be on there. Mrs. Good, what will be the hardest thing on the test in your opinion? Well, like I said, I'm more than likely going to put a couple of completion parts, but those should be relatively generic questions that y'all should be able to get. Other than that, the rest of it's multiple choice. It's kind of dependent on what you think your hardest part is. I can tell you respiration ended up being the lowest averaging test score this semester. So that probably says something. And then the next one obviously was the first biochem test. Second biochem test y'all did pretty good on. The second one was the enzymes part. The first part was organic compounds. So Carson, I would study those 2 things, respiration and the first biochem set. Okay, it's not all going to be multiple choice. I'm sorry. Sorry. Is it 22 autosomes and then the 23rd is the sex one? Yes. We have 23 total, so it's kind of like saying you have 23 pairs of socks. The first 22 are white socks. The last one's your red socks because there are some special ones. Okay, because that 23rd one, right, it's your sex chromosome, so we all have 2 of them, right? That's why it's a pair. My 2 are 2 X's, right? Legs crossed. Guys, your pair is XY for a reason. All right. Exterminate me. Okay. All right. Kind of wrapping up here on about an hour and a half worth of tutoring time. Okay, Davis. Not sure what that means. All right. Yeah. Remember, you get your small note card cheat sheet front and back only. Okay. Handwritten only. And yeah, don't forget to put your lab notebook in your backpack for your lab practical tomorrow. If you don't have it, too bad. You're still gonna take the lab practical. All right. I think pretty much done. Any other last minute questions I can answer? I don't know how you can write 10 times smaller. You're going to have a really special pen. I don't even know some of the things that y'all are saying. Can you stream Overwatch? Not tonight, yeah. And I don't play. My son plays. All right, guys. This should be able to be live so y'all can like watch it again and stuff like that. But I'm going to check out. I'm going to have my cold dinner now. And I'll see y'all tomorrow for your lab practical. All right, bye guys.